In the beginning was the Word, and the Word was God, and the Word was God.
I can hear you loud and clear.
Cool. If I have to test that mic, I'm testing it, and it seems to be working today, so we will stick with it.
Sounds good.
Cool.
I heard it was fun. It sounded good.
That I got days ago that I finally remembered.
Yeah, I set my alarm for 9 o'clock. You know, after going to bed at 6, and then
This is how bad it was. I was just saying.
Yeah.
yes. I didn't realize you were talking. I don't even have it. I didn't know how long it was. I knew how long I was sure how long it was. It's not crossword.
And then a lot of it, at least I thought.
Yeah, there.
Thank you.
Okay. Good golly, what's good to your place there? Yeah. This might be better. Oh, Digna, that's yours. That. Yeah. Yeah. Okay. I didn't use you on the curve. I used somebody else. But there's Andrew.
Yes, so Aaron, I. Oh, don't worry. Yeah, I totally get it. Thank you.
Now, do giraffes wear tennis shoes?
Huh?
Do giraffes wear tennis shoes? Do giraffes wear tennis shoes?
Oh, did you giraffes wear tennis shoes?
Yeah.
Yeah, apparently.
Oh, okay. Gotcha.
Thank you.
Selma. Jess. Okay. All right. All right. Quiz. You do not. Chicken. I did. But these were the questions that were on the Google site. No, when I looked last week, there were no questions. There were no questions there. I used what was there, so. Well, let's print it out. Might as well do it. We'll maybe make it. This will be then 2 points each. It'll be extra credit. There. How's that? Gotcha. Put your mask on, please. Put your mask on, please.
Yeah, I was just drinking.
Yeah, I know. Can you? Thank you. It's a quiz, Irma, not a dissertation. Okay, just a few words about the exam. So the curve on the exam was pretty generous. I did not include Irma in the curve. I didn't think that would be fair. So I took the top score, which was 70, and then added 30 to everybody's score. So that's your score, essentially, is your raw score plus the 30, and that's your score. In terms of what was missed after looking it over, there was not any clear pattern. Probably half of you didn't know how to draw galactose. Some of you drew mannose. Some of you drew other sugars. They weren't galactose. Some of you didn't even draw a 6-carbon sugar. Some of you draw 5-carbon sugars. That's right, you haven't. Okay, right. Maybe you'll get to draw something different. Beyond that, I didn't really see a pattern. There wasn't one question that everybody missed or that most people missed. It was kind of all over the place. So next time, I don't have access to the test banks that Macmillan has put together for the book. So I wrote all those questions myself. And yes, it took me a long time. And that's why some of the questions were probably a little harder than I wanted them to be. But be that as it may, I didn't figure that mean would be all that high on the exam. I didn't figure it would be that. But anyway, so your raw score, again, keep in mind that there's plenty of other things to do in this class, that this is not the only part of your score. So if you're highly depressed about your performance on the exam, cheer up. It's only up from here. So, okay, let's start this. And then let's go here. All right. Now, last time we saw that delta G with the little prime and then the degree sign, so-called delta G/0, otherwise known as the standard biochemical free energy change for a reaction, that it's a constant. It depends on the nature of the reactants and the products. It's not changed by the concentration of the products or the reactants or something like that. And again, this is like the delta G with the little zero, the delta G0, delta G with the degree sign, whatever, that you have in the back of your, or the inside cover of your GChem book or your PChem book or your physics book or whatever. These are measured under different conditions. These are conditions that are significant for biochemistry. Again, these numbers are not guesses. They've been well worked over experimentally. They're rather well understood. Now, there's this other value that you see in biochemistry, and it's just delta G without any of the prefixes. Excuse me, the superscripts. This number depends upon the standard biochemical free energy change. But this particular quantity is actually the delta G of a reaction at a snapshot within its progress. So this one does depend upon the concentration of products and reactants. And fortunately, we can relate these two pretty easily using this equation right here. So we have the standard biochemical free energy change plus the universal gas constant times temperature in Kelvin times the natural log of the concentration of the products divided by the concentration of the reactants. And I just have two products to reactants here. There may be four, there may be three, there may be, whatever. So just the product of the concentrations of your products divided by the product of the concentrations of your reactants. So think through this with me. What happens if this number is extremely low? What's that kind of number you're going to get here? If you don't know, pull your calculator out and try it. Go ahead. Put some really rinky-dink number in there and some really high number here, and then see what happens. You're going to get a negative number. Now, what if this number is positive, but you have an extremely low concentration of products? Could you potentially get a reaction that's not favorable to go? Yeah, you could. That's exactly how the cell is going to manipulate reactions that are not favorable. It's going to take the products and it's going to react them right away. So the product concentration is fleetingly low. That way, you take an unfavorable reaction and now you make it favorable. Because the conditions under which the reaction will proceed in the cell, I can't emphasize that enough, in the cell, are actually favorable. Because of the fact that the product concentration is so low, because it's in some sort of pathway, and that pathway does a very good job of shunting things through it. Therefore, the concentration of the products are very low relative to the concentration of the reactants. That's going to take a non-favorable reaction and now make it favorable. So that's one of the tricks you're going to see constantly. And cells are going to use this trick, and they're going to use it often. So let's just take an example. So we have this particular reaction here. This is a Krebs cycle reaction that we will go over in weeks to come. So acetyl coenzyme A is going to condense with oxaloacetate. And if we were to draw that out, it would be acetyl coenzyme A is going to react with that's oxaloacetate, and it's going to react to citrate. Citrate plus free coenzyme A. This reaction actually occurs in two steps because you get a intermediate, the citroyl. The citroyl coenzyme A is a very high energy intermediate that will then disintegrate to the two products, free coenzyme A and citrate. So this particular reaction, where you take acetyl coenzyme A and oxaloacetate, and you form a citroyl coenzyme A intermediate, that has a standard biochemical free energy change of minus 0.05 kilocalories per mole. Now, that is wretchedly close to zero. It's negative, yes, but it's so close to zero, this reaction is not very favorable. So yes, the car will start, but you had better have your AAA membership up to date. That would be a way maybe of thinking about how readily this reaction goes, if that makes any sense to you. But remember this from GChem. I'm sure you've heard this before. You'll see this again here in this class. Remember that standard free energy changes are additive. Does that also apply to biochemical standard free energy changes? Answer, you bet your bustle. So the answer is yes. So that means we can take this first reaction and its delta G/0, its standard biochemical free energy change, and we can add it to the second part of the reaction where you have this very high energy intermediate that then degrades to the citrate and the coenzyme A. That is a standard biochemical free energy change of minus 8.4 kilocalories per mole. That's a right bit more favorable, isn't it? And I see some kind of forlorn looks. Is it? This is a lot more negative than minus 0.05, right? So this is more exergonic. So it's more favorable. So we take the favorable second reaction, we combine it, or the term that's used in biochemistry, couple it to the first reaction, and we can add these two delta G's together. We get a standard biochemical free energy change of minus 8.45 kilocalories per mole for the whole thing, this reaction right there. And that is pretty favorable. That's exergonic. That's going to go. So this illustrates a second strategy that cells use to drive unfavorable reactions. Combine them with favorable reactions. So just like your car that doesn't run, when AAA comes, what do they send? They send a tow truck. The tow truck pulls up your car, and then the tow truck moves. The favorable reaction, the tow truck driving, now drives the unfavorable reaction, your car moving. So you couple the two together, just like you do with the tow truck and your dead car. Some of you are looking at me as if to say, okay, this is too close to home. Could we talk about something else? I think the illustration, though, is pretty potent, though, because the tow truck moving, towing your car, that's one's favorable, the other's not. You put the two together, and now the favorable reaction will push or pull, perhaps, the unfavorable reaction over the finish line. So this is reaction coupling. What reaction do cells use to pull unfavorable reactions? Answer, ATP usage. ATP is the universal favorable reaction that is used in cells, be they bacterial cells, fish cells, frog cells, fern cells, grass cells, tree cells, shrimp cells, sponge cells, moss cells, whatever. ATP is universal when it comes to energy in cells. It's the energy currency of cells. So the hydrolysis of ATP generates, or rather, has a standard biochemical free energy change of minus 7.3 kilocalories per mole. That's pretty favorable. So by coupling that reaction to an unfavorable reaction, you can pull that unfavorable reaction along. And that's essentially how cells are going to drive unfavorable reactions. They will use concentrations by making sure the products get used up right away, or the products are pumped out of the cell right away, or something like that. Or they will push two reactions together, one favorable, one not, making the favorable one drive the unfavorable one. Jennifer? Zero, pretty much. Once you're in the negative zone, the further you are from zero, the more favorable you get. So that minus 0.05, that was just a little tiny bit favorable. That's not really enough favorable to hang your hat on. so to speak. Now, could you make a reaction like that more favorable if you got rid of the product? Yeah, you could. But in this case, because you make a high energy intermediate, the degradation of that essentially would just pull the reaction through. So ATP uses phosphoryl group transfer reactions. So it is the energy molecule that's used in cells. If we ask why, all we need really to do is to look at the structure of it. So let's look at the structure of ATP. We have adenine and ribose. There's nothing unusual about that. But in addition to adenine and ribose, we have 3 phosphates linked together with phosphoanhydride linkages. Phosphoanhydride linkages. So these are labeled the alpha, beta, and gamma phosphates. The addition of water to this, notice that the phosphorus is going to have electrons withdrawn by the oxygens. Therefore, it will be slightly positively charged. It'll be subject to attack by the water. If you attack it, then you'll have a rearrangement of electrons, and you'll make a phosphate group that is subject to resonance stabilization. What happens if you stabilize the product? You make the reaction much more favorable, don't you? And that's precisely the case here. When you hydrolyze the gamma phosphate, you relieve electrostatic repulsion in the molecule, and you generate a product that is capable of immense resonance stabilization. Those 2 features together, plus the increase in entropy, pull this reaction forward. So you have stable end products, and you have relief of this electrostatic repulsion. So if you look at these phosphates all lined up like that, you might come to the conclusion these guys don't like each other so much, and you'd be right. So this is kind of, oh gosh, what was that cartoon with the 3-headed guy? They're always arguing with each other. It's a really dumb cartoon. I forget what it was, but you guys haven't heard of it because it's probably past your time. Remember, you? No? Okay. You didn't grow up here? Okay, yeah. Anyways, it's a really dumb cartoon as a kid. This guy had 3 heads, and they all kept arguing with each other. They argued about food. They argued about sleep. They argued about what color they wanted the bedspread to be. They argued about everything. And that's kind of what ATP, in fact, I found a picture of it once, and I used that for ATP, because that's kind of 3 phosphates all arguing with each other. You're grabbing my style. You're repulsive. You're too close. Get away. You're a bed hog. You take all the covers. You're BO. You know, whatever. And they want to get away from each other. So adding of water across this bond does exactly that. Relieves that electrostatic repulsion. And you get a product that is stabilized by resonance. Now, it is common in cells when you have ATP to stabilize it by using divalent cations, the most common of which is magnesium. Magnesium, as you see, is going to bind to these 2 phosphates. It's going to shield that electrostatic repulsion. And the substrate very often for kinases that use ATP will not be ATP, but magnesium ATP in many cases. And when you do reactions in the test tube with ATP, you almost always have to add a little bit of magnesium to make the reaction go, because this way it shields and stabilizes the ATP. Also, if you buy ATP It's almost always complex with a divalent cation, almost always. Now, that is the standard biochemical free energy change, but what about the free energy change? Given the conditions in the cell, these are all things we can measure. So you can measure things like ATP, ADP phosphate inside cells. They're a whole lot lower than the 1 molar that's used to actually derive and identify the biochemical standard free energy change. If we consider the actual concentration of these compounds in the cell, the free energy change, the delta G of this reaction, ends up being a lot more negative, about minus 30 kilojoules per mole. And that is substantial. Substantial. So ATP essentially has a lot of energy to give, and give, it does. It keeps giving and giving and giving. Here are some concentrations of these particular molecules. As measured in cells, you could see that they're pretty low. That's in millimolar, so they're way below what you'd expect for the 1 molar. Therefore, the free energy change will be much more negative than the biochemical standard free energy change for the hydrolysis of ATP in just about every cell you measure. PCR there, that's phosphocreatine. That is a substrate for creatine kinase. Creatine kinase is used to make phosphocreatine. Creatine kinase can take phosphocreatine and regenerate ATP from ADP. So this is used in muscle, and that's why it's high in muscles, low in everything else. That's why that's why creatine kinase is an enzyme that's used for muscle recovery, and that's why supplemental creatine that some athletes will use helps with muscle recovery. It doesn't do anything for strength, but it will help you recover faster. But you have to load and all this bit, and just a little word of the wise from some students who have experimented with this. If you take too much of it, it'll give you the runs. And I mean the runs. Can, yeah. You need to take it with lots of water. Lots of water. Yeah. Those sports drinks that you can get, what's the one in the green container? What's it called?
Like a liquid form or something?
Yeah, there's the guys at the weight room drink it all the time. That might be it. That might be it. Who's that really buff guy who works out like 3 times a day? I always see him there. He's always drinking that. What's his name? He throws, doesn't he?
Oh,
Yeah, Micah. Micah gulps that stuff. That apparently, when he wasn't looking, I picked up his bottle and looked at it to see what was in the. And he said, "Don't give me COVID." And I said, "No, I'm just looking at what's in it." And he said, "Is it okay?" I said, "Well, sure." There's creatine in there, not nearly enough creatine to do you any good.
I think the most I've ever had anything that I think it was like 3
Yeah.
grams.
Yeah. So it's a bit of a scam, but oh well. Sells drinks. So if we measure these concentrations and then we plug these into our delta G equation and we calculate how much energy essentially hydrolysis of ATP will generate under typical cellular, intracellular conditions, it actually comes out to minus 50 to minus 65 kilojoules per mole. Gang, that's a boatload of energy. That is a boatload of energy. And this number is typically called the delta G sub P, or the phosphorylation potential. That's how much energy you could expect from a mole of hydrolysis of ATP inside the cell under standard intracellular conditions. It's theoretical, but given that these you can measure these, there are ways to measure these fairly accurately. So while it's theoretical, because all equations are to some extent theoretical, that is a theory, but it's probably pretty close to that. Now, is ATP the only energy-rich molecule in cells? The answer is no, nor is it the most energetic. So there's one we will see when we look at glycolysis called phosphoenol pyruvate that is very, very energy rich. So phosphoenol pyruvate has a phosphate ester bond, and this is capable of extensive tautomerization. That's going to greatly stabilize the end product. Also, gang, just look at the electrostatic repulsion here. Just look at that. Furthermore, you cannot stabilize if you've got that bulky phosphate on there. So by taking off this phosphate, you yield a gargantuan amount of energy. That's not the delta G, that's the delta G slash 0. That's twice what you can get off ATP, essentially. This is a very energetic molecule. Then there's one, yeah. Oh, great question. Why do you think? Possible. That's probably it. ATP is actually middle of the road. It's middle of the road, so it can do the job, but you don't have to climb the Empire State Building essentially to make it. So that's probably why ATP essentially is the one that wins the race, because it gets the job done with the least amount of work to make it. So 1,3-bisphosphoglycerate. This has an anhydride bond between a carbonyl group on carbon number 1 and the phosphoric acid. So once again, you have extensive opportunity for resonance stabilization. You hydrolyze that, you get phosphoglyceric acid and inorganic phosphate. You're also relieving a good deal of electrostatic repulsion. Phosphoglyceric acid can then become phosphoglycerate. Remember, carboxylic acids can undergo resonance stabilization because that charge can be distributed over the whole group. That stabilizes carboxylic acids. So you got these two phosphates here, and these two are probably going to not like each other. You hydrolyze this off, you make 3-phosphoglyceric acid, you now get resonance stabilization of this product and resonance stabilization of this product. This gives you more energy upon hydrolysis than does ATP. So this is also a very high energy molecule that you can use in the cell, and it is used in the cell, as we will see. Then there's phosphocreatine, which is used in muscle. This contains a phosphorus nitrogen bond. That bond can be hydrolyzed, and it will produce inorganic phosphate capable of resonance stabilization and creatine. And the creatine itself is also capable of resonance stabilization. So it too is capable of increased stabilization. So you get more stable end products. This is more stable. That is more stable. These two, they don't like each other. They don't like each other one bit. You hydrolyze that phosphate, and now you get a molecule that's capable of resonance stabilization. This too gives you more energy than you would get from the hydrolysis of a mole of ATP. Phosphocreatine, however, can be shielded with divalent cations, and then you could use it in muscle to help regenerate ATP after extensive muscle use. Another set of bonds that are used in cells that are high energy, they're not used a ton, but they are used under certain conditions. They're used, and they're very useful, are thioesters. Thioesters. So instead of the usual oxygen, you have sulfur. And if you take that thioester bond and cleave it, you can not only generate energy that can drive a reaction, but the formation of a thioester can activate a molecule so it can then go into further reactions that don't require the input of further ATP. So if you make something like this something can usually go on to react further, and that other reaction won't require more ATP. So this essentially will store energy for further reactions. This is from your book. This gives you an idea of the energy levels of the thioester versus the kind of, excuse the expression, standard ester. Standard esters, as you could see, provide an opportunity for resonance stabilization after hydrolysis. Also, this resonance stabilization here makes this more stable. So when you cleave this, you're not getting something that's necessarily a lot more stable. This cannot undergo resonance stabilization. So this is an inherently more energetic molecule. Consequently, when you cleave that molecule, you release a lot more energy. And you'll get this sulfhydryl group here. That, therefore, can be used to activate certain molecules. And coenzyme A has a sulfhydryl on the end of it. We're going to see that coenzyme A is a carrier. When we take coenzyme A and we carry acyl groups, those acyl groups are now they're now activated for further reactions that won't require the input of ATP because that energy is stored in the thioester.
There's a couple of these molecules,
ATP doesn't have sulfur in it.
No, I know, but my question was on the extra phosphate group. So why does ATP? So you get ADP first, but you have the potential to get even more energy out of that molecule.
Yeah. And that's actually used under some conditions where you hydrolyze ATP to AMP. That releases even more energy. And then you have a pyrophosphate. You'll then use an inorganic pyrophosphatase to make two molecules of phosphate. Therefore, that prevents you from reversing the reaction. The reactions are reversible, and you increase resonance stabilization and entropy. That releases even more energy. So yes, you do have potential, and it's used sometimes. It's definitely used sometimes.
But is that not used as much because of the fact that
Right, because you don't need to. You don't need to. We'll see some examples where it is. The big example there would be activation of fatty acids for degradation. When you make a fatty acyl coenzyme A, the fatty acyl coenzyme A synthetase hydrolyzes ATP to AMP and pyrophosphate. Because you make an AMP bound acyl group as an enzyme bound intermediate that is then hydrolyzed by the coenzyme A. And then the pyrophosphate's degraded to 2 phosphates by inorganic pyrophosphatase, which further pushes the delta G into the negative range. So our old buddy here, coenzyme A, has this thiol group, and that thiol group is going to be a very important component of several different types of reactions. So acyl coenzyme A, just like what we see here, that possesses energy. If we hydrolyze it, we make acetic acid. This is then capable of resonance stabilization. That is going to generate not quite as much energy. Well, actually, yeah, pretty much quite as much energy as hydrolysis of ATP because of resonance stabilization and so on and so forth. So again, making thioesters will activate bonds for further reactions. So as you can see, when it comes to energy, ATP sits pretty much right in that sweet spot, right in the middle. And that's why every cell uses it. Very important. Very good. Let's now take a look at probably one of the most widely used set of reactions in cells, and that would be oxidation reduction reactions and group transfer reactions. Now, sometimes ATP will activate molecules by transferring its phosphate to another molecule. So you make a phosphate bound intermediate. Most of these are enzyme bound. You don't really detect them unless you use special techniques. That will activate the molecule so that it can then complete the reaction. That's an example of a group transfer activation type of reaction. So here's an example, glutamate synthetase, which catalyzes the synthesis of glutamine from glutamate. I said glutamate, I said glutamine synthetase. Glutamine synthetase. Now, notice you're hydrolyzing ATP here. Turns out the hydrolysis of ATP is going to turn this into a gamma phosphoglutamate. That is now energetic enough to react with the ammonium. That's what will make the glutamine. So you hydrolyze ATP, you get this enzyme-bound gamma glutamyl phosphate, which you can see here. Again, this is enzyme bound. You don't see it in the test tube. It's bound to the enzyme. You can put groups on here that make it not, the enzyme won't release it. You can detect it, but it's clear it goes through this intermediate. So the ATP takes its gamma phosphate, sticks it here. By sticking it here, you now make a molecule that's very unstable, but one that is highly reactive. There. So then the ammonium will displace the phosphate. Remember, the phosphate is capable of resonance stabilization, and now you make glutamine. ATP hydrolysis can also drive conformational changes in proteins. Phosphates are big, they're bulky, they're charged. when you put them on amino acids, you typically force the protein to adopt a new 3D conformation. That can actually change the modality, the functionality of the protein. There's lots of examples of this. Virtually all of your molecular machines, dynein, kinesin, myosin, all of those are going to use ATP in that fashion. Helicases, topoisomerases, those are all going to use ATP in that particular fashion. Rec A, things that move up and down other molecules, that conformational change is usually driven by ATP hydrolysis. And as we said before, ATP use is universal. Universal. Is it the biggest kid on the block? No, but it's the kid that is the most versatile. And so every cell uses it.
The example that you just gave in the previous slide, so the ATP gets hydrolyzed, but they don't have the same mechanism as the glutamine.
No, because you phosphorylate the enzyme, not the substrate. And that phosphorylation changes the conformation of the protein. In your book, there's that example of the power stroke of myosin. That's probably the best example I can think of. That's a really good example of a protein changing conformation as a result of ATP hydrolysis. So does the myosin get phosphorylated? No, but it binds ATP, hydrolyzes it, and then lets it go piecemeal to bind, power stroke, release, so on and so forth. Now this gets to Irma's question. Can ATP donate more than 2 phosphates? In some reactions, it can. Let's give you one reaction that is pretty telling, and that would be sulfur assimilation. Why do cells need sulfur? Are there amino acids that have sulfur in them? Yeah. So you need sulfur for cysteine and methionine, don't you? Sulfate does have other, no, you're right, Selma. There are other uses for sulfate. The biggie, though, is going to be those 2 amino acids. Now, in that case, your sulfur is not sulfate, it's sulfide. And that's usually not something that's available in cells. Usually it's sulfate. So you have to take in sulfate, you have to convert it to sulfide. So here's how you do it. You're going to use an enzyme called ATP sulfurylase. ATP sulfurylase. So this will take ATP and it's going to stick the sulfate on the ATP, but it's going to hydrolyze the ATP to AMP. So here's how this reaction goes. You take ATP and sulfate, you encounter this enzyme. This is a pretty universal enzyme. You take the enzyme from bacteria and from people and from dogs and cats and fish and flies and whatever. Those enzymes have a fair amount of similarity. You're going to clip the bond between the alpha and the beta phosphate. You'll generate pyrophosphate. This will then get hydrolyzed to 2 phosphates. You'll stick that sulfate right there. Now, essentially, you've made this molecule called 5-phosphosulfate, adenosine 5-phosphosulfate, or APS. APS. That's the first step in sulfur assimilation. Then there's a process called adenylation. Say that 5 times fast. Adenylation. I told somebody once, use this at a party to show everybody how smart you are, and they said, look, after a couple of drinks, I'm not going to be able to say that. Anyway. In this case, you're generating AMP, and AMP gets attached to something. The AMP gets attached to something. That's called adenylation. Yeah, Gesundheit. So, fatty acid oxidation. You have to activate that carboxylic acid group of the fatty acid. So you're going to take a fatty acyl coenzyme A synthetase. There are many different types of these. They specialize for different length fatty acids. You're going to take ATP, and you're going to clip it between the alpha and beta phosphate. You'll generate AMP and an organic pyrophosphate. You make an enzyme-bound intermediate that now has AMP bound to the acyl group. That is easily displaced by coenzyme A. And now you form a thioester between coenzyme A and the fatty acid. That now prepares the molecule for beta oxidation, which occurs in the mitochondria. And then that inorganic pyrophosphate is degraded by inorganic pyrophosphatase, further pushing the reaction into the negative delta G zone because you just made it irreversible. And here's essentially how this goes, and we'll probably end with this. Here's your fatty acid. This is palmitate. Palmitate reacts with ATP and fatty acyl coenzyme A synthetase. You clip this here. You attach this to the fatty acid. That's a fatty acyl AMP. Does that look stable? Survey says, niet. So coenzyme A comes in. You have a sulfur. Sulfur, remember, has extra electrons. It looks at this poor electron-deprived carbon with a lean, with a lean and hungry look. It attacks there, and it displaces the AMP. And now you make a thioester. This is now prepared for further reaction, and away you go.
So why would it have to cleave both of the phosphate groups? Is it because the intermediates would be more stable that way?
The intermediate is more reactive. It's more reactive for the reaction with coenzyme A. Yeah, that's the price you pay for having it react with coenzyme A. Because reacting it with coenzyme A now means the rest of the reaction, you don't have to add any more energy to it. It's ready to go essentially straight into beta oxidation. Yeah. 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 All right, we'll pick this up next week. Have a great weekend. See you all later.